Okay, so we're going to look at the cell membrane briefly. I'm going to do the screencast looking at the structure of the cell membrane. So all we're going to do is look at the parts, and tomorrow morning in class, we'll go quickly through a diagram that'll help you to label so that you get comfortable with those structures. But then our real focus and the stuff I want to spend time in class on is going to be on the function of the cell membrane and transport through the cell membrane. So that's the part I feel like you need kind of to be in person and be able to ask some questions for. So learning goal, you should be able to describe the structure of the cell membrane according to the Fluid Mosaic model. So you should be able to explain to me what a membrane is made up of. You should be able to label a diagram. You should be able to describe the function of the membrane and explain why the term the Fluid Mosaic model is appropriate. So a biological membrane, as we talked about earlier, they're essential to life. Sort of in the evolution of things, we had small organic molecules that were essential to life, and then we had macromolecules forming. We had those macromolecules being engulfed by an assembly and then being able to direct the replication of that assembly. So these membranes really are a crucial sort of part of life. And they're important not just in surrounding individual cells, but also in terms of surrounding organelles. And that's actually the definition of an organelle, is that it's something that's membrane bound. And membranes are very active structures. They're composed of lipids, proteins, and carbohydrates, and they help maintain the internal environment of the cell separate from the external environment. So the membrane has to be able to transport raw materials in, transport manufacturers and waste out, prevent the entry of unwanted material into the cell, so keep viruses, for example, out, or something that could be toxic to the cell process. Prevent the escape of matter needed to perform cell function. So cells work really hard to get lots of glucose and sugars into them. You don't want them to just sort of ooze out naturally. So this is an early picture of cell membranes. So these are actually two cells side by side. You can see this dark line is one membrane and this dark line is another. But if you look up close, what you'll see is that what appears to be one dark line is actually two. So those two dark lines sandwich an inner lighter core. So you're actually seeing two cell membranes here. So this animation looks at the fluid mosaic model. You can see the outside of the cell, and the cell membrane is composed of a phospholipid bilayer. The phospholipid bilayer, these are my phospholipids. So they have a charged head, which is hydrophilic and will interact with the water on the outside or inside of the cell, and nonpolar tails making up the inner portions of the membranes. They also have proteins embedded or associated with them. If the protein is like this one, a through and through protein, we call it an integral protein. So those might be things like ion channels and so on. These proteins that don't protrude straight through the cell are called peripheral proteins. And we can have attached to those proteins carbohydrate chains or oligosaccharides embedded. The one thing I did want you to notice when I had the animation playing is that things were moving and floating around. So we don't get a lot of movement of phospholipids from layer to layer, from top to bottom, but we do get a lot of movement of phospholipids within the layer, which is why the bilayer is termed fluid. And that's important because it helps in membrane transport, it keeps the cells from freezing, and so on. And part of that fluidity is maintained by the presence of cholesterol molecules. So I said when we were talking about lipids, even if you don't eat fat in your diet somehow, which is impossible, you would still have to make them. You'd have to make cholesterol because you need this in your membrane, particularly at extreme temperatures. So all of these different components are sort of flipping and flopping and flowing around in that membrane. So cholesterol, peripheral proteins, integral proteins, and then oligosaccharides. And the oligosaccharides can be attached to a lipid or they can be attached to a protein. Usually those play a role in cell recognition or cell signaling. So early understanding of the cell membrane, I'm going to do the same thing I did with the Darwin note earlier. So we're going to look at, these are the observations, and these are the conclusions or the inferences that were made from those observations. So in the early 1900s, researchers noted that lipid-soluble molecules entered cells more quickly than water-soluble molecules. So that led scientists to believe that the membrane probably consisted of lipids. And by 1925, they could confirm that those were phospholipids. Early electron microscopes showed that image that we looked at on the previous slide, that sandwich-like structure. So that suggested that the phospholipids were arranged in two layers, what we call a bilayer. And we also know that water-soluble molecules can still make it into the cell. So there must be a protein or some sort of structure that allows water solubles to make it through the nonpolar lipid tails. So we call that a phospholipid bilayer, and then the proteins inserted into the bilayer ensure that polar molecules can make it through. So the proteins that move through the bilayer have nonpolar regions that are in contact with the nonpolar hydrophobic fatty acid tails of the phospholipids. And then the polar portions stick out of the membrane into the water, into the intercellular fluid. And you'll also have like a polar core that will allow those things to pass through. And because all those molecules are held together by sort of hydrogen bonding and nonpolar interactions, the attractions are very weak. So the bilayer is quite fluid, and the phospholipids, as you saw in the animation, are constantly moving laterally across the membrane. So they don't flip very often from top to bottom, but they move within their layer quite easily. And because those other molecules are randomly sort of peppered into that fluid, that's where the term mosaic comes from. So if you haven't heard the term mosaic before, this is a mosaic. If you go to St. Pius, they used to have mosaics of all of the Stations of the Cross along the side wall. But it's basically where one big picture is made up of lots of tiny stones that come together. So similarly, the overall cell membrane is made up of lots of different components, proteins, peripheral proteins, integral proteins, oligosaccharides, all in a whole. The features of the fluid mosaic model, we call each membrane a leaflet, each layer, and it acts as scaffolding for the other components that it needs. And its fluid structure is important because if there's a puncture or a tear in the membrane, it immediately moves to seal itself. So the role of cholesterol, it maintains membrane fluidity. I'm going to go back a few slides. And then back, ahead, back. Hold on, I'll show you on the next slide, hopefully. What happens is cholesterol inserts itself in between the fatty acid tails in the membrane, kind of pushing them apart. So at room temperature or higher, it's another source of hydrophobic interactions in the membrane, holding them together. So at high temperatures, the cholesterol interacts with the fatty acid tails and sucks them in together. At lower temperatures, it pushes them apart so they can't solidify. And the proteins, we talked about integral and peripheral. They stabilize the cell membrane and often can form anchors to the cytoskeleton, which itself is protein. And they can form a whole bunch of different roles: transport, catalysis, cell recognition, signal reception, and transduction. So you can have receptors on the surface of your cell, which are going to detect other compounds. and send out a signal in response. Here's the structure. This is what I was looking for. So you can see, here's my cholesterol, and at high temperatures, this cholesterol is going to interact with these inner fatty acid tails and help hold those lipids together. At low temperatures, it's going to push them apart, keep them from getting too close together, so they aren't able to freeze, basically. So here we've got our carbohydrate chains on our proteins. We can also have carbohydrate chains attached to lipids. So this is called a glycolipid when it has that. These can be involved in cell recognition. So your immune system, for example, is taught to recognize the glycolipids on your cell as perfectly normal and not worth attacking, whereas the glycolipids on a bacterial cell, it's going to recognize as foreign, and that will be its signal that that bacteria needs to be attacked or invaded. Glycoproteins can be involved in cell-cell signaling, or again in that cell recognition piece. These integral proteins that move through and through could be channel proteins and carrier proteins, which we'll talk more about tomorrow, that are involved in transport, or they can be involved in a signaling pathway. So sometimes something can bind to this protein outside the cell, and it will trigger a series of shape changes inside the cell that causes some reaction to happen inside. So a hormone, for example, would bind to a protein on the surface of a cell, trigger a change, and cause some effect inside. The cytoskeleton is connected to the proteins in the cell membrane. Its job is to support and anchor the cell, as well as providing shape and motility. And it's made of microtubules, intermediate filaments, and microfilaments that are made of proteins like actin and myosin. And it can connect to cilia and flagella. We talked about the role of carbohydrates. This is the very blurry model that you're going to get in class. So this is where we'll pick up tomorrow.